Okay, I will start then, and I disappear.
Shape the future with E-Series.
Le grandi conquiste si fanno insieme. Intellimec, il consorzio intelligente per la meccatronica.
Good morning. My name is Cecilia Lasky, and it is my great honor this morning to introduce the plenary lecture given by Professor Barbara Webb. Barbara Webb is a professor of biorobotics at the University of Edinburgh, and she has a PhD in artificial intelligence, and she proposed a new approach of using embodied robot models, not just as by inspired robots inspired to living systems, but using this robot model for explaining and for studying living beings. She's really a pioneer in this kind of biorobotics, where robots can help studying living beings and represent embodied models of them. And she has been studying especially the insert sensory motor systems. As a professor of biorobotics, she's continuing this study, but she has moved her research towards more complex insect behaviors like learning and navigation. And this is what she will tell us this morning in her plenary lecture. I just want to add one small thing. I said that she's a pioneer in the application of robotics in the study of biological system, and she actually wrote a seminal book. At least it was a very important book for me, called Biorobotics. It was, I think, the year 2000, if I'm not wrong. So again, my great pleasure to introduce Professor Barbara Webb. The floor is yours, and again, her lecture is about insect inspiration for robot navigation. Thanks a lot for accepting to be here.
No problem. I will just share my screen. Hopefully that's clear. You can see my slides. Yeah. And can I just check? Can you see my mouse as well? Yeah.
Yes.
Great.
Perfectly.
Good. Okay, good. So thank you very much for the invitation and the opportunity to speak to you. As I said in the chat, I would have loved to actually have been in Italy. I love Italy. I've spent some time there. But nevertheless, I hope it will be an enjoyable event. So I'm going to talk about insect-inspired robot navigation today, and I'm going to go straight on to the insects that I'm inspired by. So this is one example of an insect navigator. It's a desert ant, and what you can maybe see is it's running home very quickly, and what it is not doing is it's not following a chemical trail. It's not encountering any other ants. This ant is foraging completely individually. And these ants, basically, because they live in the desert, it's too hot to leave chemical trails. They would just burn off. And they have to forage very quickly because it's so hot, it's actually dangerous for them to be out there. So good navigation is crucial to their survival, and it's crucial for the colony because they have to bring food back to the colony. These ants have been observed to do this behavior over distances as large as 1 kilometer away from their nest, where their nest is just a small hole in the ground. And they can do this, as you can see here, in quite complex, cluttered terrain or in very barren areas as well. And there's a couple of key mechanisms they use, which are path integration and visual memory. I'll talk a little bit more about each of those. But first, I just want to sort of give you a flavor of the field studies we actually do with these ants. So this is work that's been done particularly with Michael Mangan and Antoine Wistra. And this was one field season where we actually followed the ants around using differential GPS so that we could track where the ants were going. So it looks a little bit like this in the field. We have an ant nest, and we let one ant out and we follow that ant and then wait for it to come. We follow its whole path in this, as you can see, quite cluttered kind of area, and then we wait and follow it back home again. And while we're doing that, we'll also have a downward facing camera, so we can actually try to track in a little bit more detail what the ant is doing. And this is tracking work that was done by my postdoc, Benjamin Risser. Really impressive that he can track an ant moving through while the camera is moving through this kind of undergrowth. Really super tracking system so that we can get some quite detailed information about what the ant is doing at any point in time. And the other thing we did on this particular field study was we actually took LiDAR scans so that we could reconstruct the world. And then here I'm showing you this world from the ant's point of view. And the point here is, is that the ant, let me just show that one again. The ant has quite low resolution vision. So what it sees is, this is about the resolution, maybe even this is slightly higher resolution than an ant actually experiences. So this is what that world that I just showed you looks like from the ant's point of view. So it can navigate around this world. And as I said, there's two main ways that the ant does navigation. So the first one's in biology, we usually call it path integration. In robotics, it's probably more commonly called odometry, but it's the same principle. You're keeping track of your own movements so that you can calculate your way back home. And in this case, this was an ant. It was the very first time this ant had ever come out of the nest. And we followed her from the nest, and she came out on this kind of complex route that's sort of shown in orange here. And all the way along that route, she's integrating her motion, her direction and distance that she's traveling, so that at the end of that route, when she gets some food, she can go straight back to the nest. And these blobs are some of the vegetation, so she can't see the nest at all from this position, but she nevertheless can go pretty much straight back to it. So she not only goes in the right direction, but she also knows the distance. She also knows the distance because when she overshoots, she turns and comes back and finds her way into the nest. And it's been known for a very long time that ants are capable of doing this. One thing that might come up again later, most people who have done robotics know that pure odometry tends to accumulate error, but ants use an external compass, which is the polarized light in the sky. They actually use that as an external compass, and this can make odometry substantially more reliable than it might be if you're just using proprioceptive cues. Okay, then the second main mechanism that ants use is visual memory. So this is an example of one of these ants that we followed multiple times to and from the food. So if they find food in a particular place, they will nearly always go back to the same place to find the food again, and they'll just go over and over. And each ant, in this case, will establish its own particular route outwards to the food and back again to the nest. And really, I mean, they try and go, they're trying to go roughly straight, but because of the vegetation, they have to go in some kind of route around the vegetation. And a different ant might just happen to go on the other side of a particular piece of vegetation and establish a slightly different route. But once they've done a particular route, they will be very, very consistent. So this is about 10 tracks of the same ant over several days. So it comes out the next day and it follows the same route again. And once they have established this kind of route, then they actually no longer need the path integration. So for example, if you pick up the ant from the nest and put it halfway along, when it's at the nest, it should have 0 for its path integration, but you put it on its route and it's perfectly able to follow the route back home again, even though its path integration says it should be home already. If you pick it up and put it somewhere off the route, here it's, this again, it thinks it should be at its nest, so it does a search pattern. But then as soon as that search pattern crosses the route, it will follow the route back home again. And again, this is a behavior and capability in ants that's been known for quite a long time. And we know that lots of other insects can do this as well. Bees in particular show both of these. They can do path integration and visual memory, use visual memory as well. And so can wasps. And I think people are slowly showing that other insects are capable of it also. Okay, so what I'm interested in is how does the ant do it? And maybe a bit more specifically, I'm actually interested in how the ant's tiny brain is able to do this and how we can copy it for robot navigation. So I think it's really striking to compare insects versus something like self-driving cars. So self-driving cars have been amazing. It's been a huge success for robotics, what cars can do, what autonomous cars can now do. But nevertheless, they can do it partly because they have conventionally structured roads, and we supply the car with a really detailed 3D map, usually from large-scale advanced data gathering on the region that we want the car to be able to drive around. Whereas insects are capable of dealing with entirely novel, unstructured terrain, uneven ground, vegetation, without any particular prior knowledge. So if you take a bee's hive to a completely new location, they just do a couple of learning flights, and then they seem to be able to navigate incredibly well in that arena. Secondly, self-driving cars, one of the reasons they can do what they do is that they really leverage high degrees of sensing. So typically they might have multiple high resolution cameras. In particular, they use a lot of range sensing, laser or radar range sensors. They also exploit very accurate inertial motion systems and odometry as before. And then also they use external cues such as GPS and wireless. And in fact, autonomous cars wouldn't be possible if it wasn't for the whole GPS satellite system. Whereas insects don't exploit any of these things. They mostly use vision, and their vision is actually quite low resolution. So this is just a close-up of the eye of one of these ants. And you can see the ommatidia. Each of those is equivalent to a pixel in vision. So it's really quite low resolution. But what is notable about it is that it has different properties to our vision. So in particular, they're sensitive to ultraviolet light, and they're also sensitive to the polarization plane of light. So I'll end up talking a little bit about both of those. They also, of course, have other sensors such as chemical and tactile sensors and proprioception, but I'll be focusing on vision for this talk. So we were quite interested just in this idea of, well, why do they have ultraviolet vision? And one hypothesis was that this might help you to tell the difference, to make a sharp distinction between the sky and the ground. And I'll come to in a moment why that might be useful. But we just did some testing. We just took a conventional camera, but took out various filters and so forth and put in other filters so that we could actually use it as a UV camera. So we could take pictures either in the normal RGB visible range or in the UV range. And this is a picture in the ant field. And you can probably see that the sky is clearly a different, has a lot more UV in it, so it's more intense. This is a picture in Edinburgh. And again, the UV gives you a very clear, and what you can see down the bottom here is if we try and kind of separate the sky pixels from the ground pixels in RGB space, there is separation. You can separate them in this kind of three-dimensional RGB space. You can see there's a cluster for the sky and a cluster for the ground, but there's quite a overlap between those two. So there's a number of ambiguous points. Whereas if you do this just with the single UV channel, then as you can see, there's huge, very, very clear separation between sky pixels, which are the high UV, and ground pixels, everything else, which is low UV. So why is it useful to extract the sky versus the ground? Well, you can actually use this quite well to recognize things. And I'll come to that in a moment, but I also want to point out that actually it's also using the UV is actually very robust to changes of time of day or the weather conditions. So we wanted to test that with kind of changing weather conditions, and anyone who's been to Edinburgh will know that you get a lot of different weather conditions in a very short time, often in Edinburgh. So this was all on one day. We just took pictures of Edinburgh sort of as it went into the evening. And we did the same thing in UV. And you can see even though here it looks like you can't see anything, in fact, the sky part is very slightly still more intense than the ground part. And then if we try to say, well, let's try and classify pixels as the same or different, sky versus ground, then it was very, very easy to do in UV. And we basically got perfect classification. So every time we classified over multiple pictures at multiple times of day, multiple weathers, it always made the same classification, whereas trying to do it with typical visible light, you could see that the clouds really mess things up. And then there's also often a lot of stuff in the ground that is similar to the stuff in the sky. So it's quite difficult to do. Okay, so what can we use this for? So what we did was to actually then take our camera and point it at a mirror so that we could get a kind of omnidirectional picture, which is what ant eyes have. They have quite a 360-degree view of the world. And we filmed as we walked in Edinburgh. And this is what it looks like. I hope you can see that. I'm just getting a message that my connection was unstable. So I hope we're not missing something. So this is segmented by the UV, and hopefully what you can see is that we can basically see that this is the shape of the sky, of the ground against the sky, the buildings, and then here we go through a park. So this is the trees against the sky. And it turns out this shape is actually extremely recognizable. So it's a very simple binary image, but if you just look at that shape, you can pretty much recognize anywhere that you are on this route just by comparing that shape to the shape on the route on another time. So we actually did this route a couple of times at different times of year even, and we could recognize the same places on the route just by this two-dimensional sky shape of sky versus ground. And this is just showing another application of this. So this is, we actually tried to say, can we actually, that shape could be displaced if the camera isn't pointing exactly upwards. So for example, if you have a robot here that's going over uneven terrain, but nevertheless looking up at the sky, so this is again RGB camera and a UV camera, you see this canopy pattern, and it's actually quite distinctive. And if you again do the simple UV filtering with a threshold, you get this very distinctive pattern, and this pattern is actually quite recognizable. So even though it's tilted or shifted, you can still recognize very well where you are. So we actually used sequence slam, which is a classic kind of place recognition algorithm, and showed that you could do much better with sequence slam if you use the UV sky segmentation. Okay, so that was work we did a little while ago. So for the remainder of the talk, I'm going to actually talk about some of the brain mechanisms behind this. So again, if we compare self-driving cars to insects, self-driving cars need powerful computers, and this is actually a few years old now, but it was an example of, NVIDIA were releasing something that could do 24 trillion deep learning operations per second, which was designed specifically to be used for autonomous driving. Whereas insects have a brain that's less than the size of a pinhead, with about 100,000 brain cells, and each brain cell probably has a processing speed on the order of 1 millisecond. So there seems to be a huge mismatch in the computational power we have available here. So it's clear that insects must be doing something pretty efficient with their brain. So this is something that we've basically been interested in for the last 10 years or so in my research group, is how does the insect brain support navigation? And this is just a sort of picture. This is actually a fly head, but it sort of shows you the insect brain. And this is, for example, the optic lobes. So you can maybe see that there's a lot of visual processing going on, even for a fly. And then there's some specific areas in this brain that seem to be quite important for navigation, or we hypothesized would be important for navigation. So the first one is, I talked about the visual memory, and to do visual memory, basically what you have to do is do pattern recognition. And although not much was known about visual pattern recognition in insects, we did know that olfactory pattern recognition was done in this area that's kind of in blue here, and this is kind of blown up here, an area which is called the mushroom bodies, basically because it's shaped like a mushroom. That's the this set of neurons. So we thought that might be a good place for the visual pattern memory. And then similarly, for path integration, we know that an important ingredient of path information is to know your heading, to have directional information. And we know increasingly more about an area of the insect brain that seems to act like a compass. And that's this area right in the middle up here, which is being blown up here. And it's called the central complex, basically because it's in the center of the brain and it has a complex architecture, which I will describe. So essentially, we were trying to kind of bridge the gap. And part of the problem is it's quite difficult to, for example, record from these brain areas while an insect is actually doing real navigation in the wild. So we didn't have any kind of direct evidence that these brain areas were involved in navigation. So our strategy was to test models of these brain areas, so to build computational models of these brain areas, and then to test whether they could actually support navigation on robots. So I'm just going to kind of quickly describe the models that we built of these two areas, probably not in enough details, so people can either ask questions or, again, you can find the papers, but it gives you a flavor of what we try to do here. So let's start with visual memory in the mushroom body. So as I said, visual memory must involve pattern recognition. And this just kind of gives you an idea. This is a little simulation now. This is actually it's exactly the same vegetation pattern that I showed you sort of earlier, but we've now just kind of put it into a little simulator so that we can simulate what it looks like for an agent that's moving through the world. So here you can see this is the low resolution vision that the agent has. And what we're assuming is that what we want it to do is to remember each of these visual patterns. So as it goes along this route, there's about 100 visual patterns, and it should just remember what those are. Okay. And the reason you want to remember those is because you can then exploit this quite neat idea for following a route again, which was actually first proposed by Bart Baddeley, which is that you have at any point along the route, so this is sort of the route that you've run along for 8 meters. At any point on that route, if you stand on the route again and you look to your left or you look to your right, so that's what's sort of shown along here, the heading angle, then if you're looking in the same direction as when you went along the route in the first place, and you compare what you currently see to the image that you stored, you should get a 0. Okay, it will be exactly the same. So this is the image difference on the z-axis. And as you turn and face a little bit more to the left or to the right, the match will get worse. And then the same happens all the way along the route. There will be one image in your whole set of images. So you always compare what you see now to everything that you stored. But if you're looking in the same direction at the same place as you were before, then one of those images will match. And if one of those matches, then that's the direction you should go. If nothing matches, you should turn around until you see something that does match. So essentially, you can follow this kind of valley of image similarity all the way back home. So this is the kind of algorithmic concept, but we wanted to do this in the brain of the insect. So the concept is that the ant wants to associate all the views that it experiences as it goes either towards the food or towards the nest, and to kind of just associate those experiences with the fact that it's going in the right direction. And as I said, the area of the insect brain that seems to be specialized for pattern learning and has been studied mostly for olfactory pattern learning, called the mushroom body, in ants, we know it has lots of visual input as well. So this is the model, and I'll just try and give you a kind of broad feeling for how it works. As I said, people can ask if there's more questions, but the basic idea is that you have sensory inputs. So as I said, this was originally an olfactory model, a model of olfactory processing. So you have olfactory inputs, but we assume for the ant it also has a number of visual inputs. And here we're really just mapping pixel by pixel straight into this circuit. And the mapping is expansion mapping. So basically we have a smaller number of projection neurons and we go to a large number of these neurons, which are called Kenyan cells in the mushroom body. And in the ant, the ratio between these is, maybe something like 1,000 to 100,000. So it's a huge expansion into the Kenyan cells. And what appears to be the case is that any pattern in the sensory neurons is projected into a sparse pattern in the Kenyan cells. So just a few Kenyan cells will be activated. There's kind of a random projection pattern that means that, whatever view you happen to have here becomes some sparse pattern across the sensory neurons. And then all you need to do to remember that pattern is to just associate every time you see a particular pattern and you're, for example, going home, you need to just change the weight of those few neurons that are active. You need to change their weight to this output neuron. Okay, so that if you get the same pattern again, you can recognize that you have the same pattern again. And you just, all the views you want to learn, you just put each one on top of each other. So your output doesn't tell you which view you've seen, it just tells you this is a view that I have seen. Okay, so any view that's completely novel pattern up here will give you a different output to any view that you've actually actively stored. And you can maybe, here we've kind of illustrated that you might have a different set of memories for the going home and for going out towards the food, for example. So that's the general idea. One nice thing about this is that it very easily can be made into a multimodal sensory system, because you just add in whatever other senses you like and you just learn that pattern as well. So it means that rather than just recognizing that a place is familiar in the sense that you can, you've seen it before, it might also be familiar because it smells the same or the wind is coming from the same direction. So it's a kind of a gestalt memory of familiarity of a place. And it turns out this works quite well for visual homing. And we can also actually calculate what the memory capacity will be. And then maybe just in the interest of time, I'll skip over this. But what we could do is we could actually come up with an analytical solution for how many visual memories the ant should be able to store, given the assumed sparsity of the code and given the number of neurons in its in this mushroom body. And it turns out, certainly with very reasonable assumptions, it could easily store more than 400 images. To do this, we actually, the original model was actually a spiking neuron model, but we simplified it to a kind of binary abstraction, which also allowed us to implement it efficiently on a robot. So this is just to show you this principle at work on a robot. This was our very cheap and cheerful robot, which is a mobile phone on wheels. And here it is in its little kind of simulated ant environment. So we just put a omnidirectional mirror again on the camera. So this is the phone camera, has a little omnidirectional mirror so that we can see the world around. And again, we store it in this very low resolution form. And then at any point, we either, if what we see matches something that we saw before, we keep going. And if it doesn't match, we will oscillate back and forth and try and get a better match. And we also took this robot back out to the ant field and tested it out in the ant field as well. And it was able to follow the route that it had learned. Okay, so with the little bit of remaining time, I'm just going to talk now about path integration in the central complex, which is the more recent work that we've done. And this is work that's been done in close collaboration with Stanley Heinze at Lund, who's done all the biology work that I'm going to describe, all the circuitry, and Tom Stone, who's a really good PhD student with me, who's now at Google. And what we wanted to do is understand whether this amazing spaghetti of connections in the central complex could actually function to do path integration. So it looks like spaghetti, but in fact, there's a lot of structure in here. So for example, there's a set of 8 columns across this area, which is called the protocerebral bridge. And there's another set of 8 columns in this area, which is called the fan-shaped body. And in fact, these connections where you can see the neurons going across, it's actually a very, very consistent pattern of connections. So I'll talk a little bit about that as we go along. So the reason why we thought this is where we might find the path integration circuit was firstly because we knew that the compass information that the insect uses goes into this area of the brain. And this was quite old work that Stanley Heinze had done. So we know that the sky has a polarization pattern. So this is just kind of, if this is where the sun is in the sky, there's a pattern of polarization caused by the sun. And if you kind of mimic that here with just a simple polarizer that has a particular direction of polarization, and you record from neurons in this area of the brain and you rotate the polarizer, then what you see is that the neurons respond specifically to a specific direction of polarization. And what's more, there's a very regular map. So as we go across these columns of this area, each column has a different preference for a different direction of polarization. So it seems like we really have something that's like a good compass input. And then more recently, Stanley also found that we have speed input to the central complex as well. So in this case, it's in the form of optic flow. So he was recording from this particular bee, and the bee is sitting, being recorded, with a brain electrode in its brain, but you show it a video, basically either of the world moving past it very fast like this, or moving backwards the other way, or rotating. And he found, again, there's some specific neurons that were particularly sensitive, in this case, to front to back motion. They didn't really respond, or if anything, were slightly inhibited by back to front motion, and they wouldn't respond to rotational motion. And in fact, these neurons would respond more and more strongly the faster the simulated forward flight speed was. So it really looks like they were encoding speed. However, one slight mystery was when he tested to see, what's actually the optimal cue for these speed neurons. And you would think that it should be, when the world is moving from the front to the back, that should be the best cue. But it turned out that there's one of these neurons on each side of the brain, and they actually had a preference not for straight ahead, but for movement at 45 degrees. So I've kind of tried to illustrate this here. So the best response of this neuron was for a bar that either moved in this direction on that side of the visual field, or this direction on this part of the visual field, but this changeover was not at 0, it was at 45 degrees. And then for the other neuron, it was at 45 degrees the other way. And it wasn't clear why this would be a good idea, but we worked out, and probably Tom should be credited for spotting this. What this actually does is to allow the animal, in this case a bee, so it's a flying animal, to measure its actual velocity, even if it's not flying in the direction that it's facing. So if you imagine an insect flying, it might be side sweeping, it can move in a holonomic way, and we want to represent the actual velocity rather than just the velocity in the direction that it's facing. So if you imagine here, this is the direction that its actual head is pointing, but it's actually flying, flying slightly off that direction, so it's got a slight side slip. Then because it has these two neurons which are sensitive at 45 degrees on each side of its heading, what that means is that its actual motion is being projected onto this orthogonal basis of the preferred motion of these two neurons. So the neuron in the left part of the brain will be more responsive, have a stronger response than the one in the right side of the brain. So between them, you're actually encoding the real velocity in a geocentric framework. And this is what you want to do. If you want to do good path integration, you actually want to know where you are moving in a geocentric framework. So you have the polarization information from the sky, so that's in a geocentric framework, and you have this motion information on relative to the ground in a geocentric framework. So between those two things, you can actually do quite accurate path integration. So the hypothesis, again, we won't get into all the details of the circuit here, but basically what Stanley discovered was that there were specific neurons in the middle part of the central complex that get both these inputs. So they get the input from the compass, they get the input from the speed neurons. And so all you need to do is integrate those two things, and you potentially can end up with a path integration memory. It's actually in a kind of distributed form. So you have one neuron for each heading direction, you have one neuron that's accumulating for that direction. So across the set of neurons, you have the actual vector that you're going in. So that was our first hypothesis. But not only did we think we could get the path integration to happen here, but also that we could understand how the animal could use this information to steer itself. And so that's the second part, which again looks quite complicated. So this was this kind of circuit diagram that Stanley had come up with over years of experiments and neuroanatomy for what happens. So this is kind of that spaghetti that I showed you before. This is the organization of the spaghetti, but it still looks pretty much like spaghetti. But one of the things that you can kind of see here, and we'll see it more clearly in the next picture, is that there's this point at which the information kind of seems to get shifted across by column. Okay, so instead of just going straight down from one column to the next, you get this kind of cross connections. And again, not to get into the nitty-gritty, we tried to say, well, if we take all these connections, which are all determined purely by anatomy, so this wasn't devised, as an algorithm, this was just anatomical connections that really exist in the animal. And we put all these connections into a force graph, where the idea is that you want to show what's the actual underlying structure of this connectivity in the most efficient way. And quite amazingly, this is what comes out of that force graph. It's a perfect ring, which is not, it's not obvious from the previous picture that this would be a ring, but in fact it does close into a ring. And it's just beautifully evolved to do exactly what we want for path integration. So very briefly, I'll sketch the idea, but again, it's just such a nice circuit that I really encourage you to go have a look at the paper. So this circuit basically has an inner layer, which is the compass. And then that connects to these neurons, which we're calling the memory cells, which get input from the speed neurons, and they accumulate. Each of them kind of accumulates in the direction of that compass. But there's two of them, and one of them connects to the right output neurons, and one of them connects to the left output neurons. And what they do in that connection is they compare the memory that you've stored up to the current heading, either the current heading to the left, or for this case, the current heading to the right. And what that allows you to do is, if you want to go in the direction that you've stored in your memory, then what you want to do is turn so that your current direction matches better that memory. So by comparing it to the left or to the right, you can work out, should I, at this point in time, should I turn left or right to align myself with the memory direction that I want to go. So let me just show a little simulation of this happening. So this is a little simulated bee flying along. At any point in time, the compass heading is encoded by this ring, you can kind of see, and it actually accumulates in the opposite direction to which it's going, because it wants the sum of all these memory neurons to basically point back towards the nest. So it's gradually getting stronger and stronger in this direction. And then when we want it to go home, we let the steering circuit take over, and it steers it straight back to the nest. And because we don't have any stopping criteria here, what it will do is just actually spontaneously produces this search pattern, because the memory will get balanced and then it will go a little bit away and that will be unbalanced and that will bring it back again. So we actually get search for free out of this. Okay, so let me just very quickly say we've now put this on a flying robot. So this is work by Jan Stankiewicz that we just published this year. So we have a downward facing camera, and with a gimbal to kind of stabilize it in the way that flying insect heads seem to be stabilized. And we can put the whole circuit on the computer on the flying robot, and here it is. And we're using this optic flow to estimate the ground speed. So we're looking down at the ground and we're filtering for a preferred direction of motion at 45 degrees plus or minus, like I described. And we can use that to estimate our speed fairly accurately, but it is quite important to have the head stabilization. So if you don't have this gimbal to stabilize the head, it's much, much noisier. And we can put this on the robot. And this is the robot actually flying out on a kind of L-shaped path we also put oscillations in the path. So it's gone about 100 or more meters away, and then it uses this path integration circuit to get itself back home again. And when it comes home, we can measure the offset. So this is kind of the error that's accumulated with the distance flown. And what you can see here, hopefully, is that it's actually the error for, even 150 meters is still only a few meters away from its starting position. And we've tested this both in the real world and then in a number of kind of simulated worlds where we tried to make things more difficult for the system. And I think I'll skip that. So let me just conclude. So why are insect brain circuits relevant for robotics? So I think one reason is that if you think about insects, they have the most amazing variety of body plans, of locomotion capabilities. Some of them fly, some of them crawl, some of them burrow. They live in every possible ecological niche you can imagine. Yet the brain structures that I've just told you about, the mushroom bodies and the central complex, occur pretty much across every single species of insect. So it seems like they support some kind of key general functions that really work for adaptive behavior, and they've hit upon some circuits that really just do what you need the circuit to do things like navigation. So, for example, the mushroom bodies are especially well developed in insects that can do complex navigation and complex memory tasks. The central complex is especially well developed in insects that have some kind of complex motor coordination and highly directed motion. But nevertheless, both these circuits have orders of magnitude less complexity than human brains or than many current robot algorithms, yet seem to be able to solve some quite comparable tasks. So that's why I think they're really worth study. So essentially, we can use some of these ideas from insects to find low-cost, low-computation solutions for robot navigation. But at the same time, we can also use the robots to test our hypotheses about the brain circuits that support navigation. So I'll finish there and just say thanks to the people who helped do this work, particularly, so these are all the people from my lab who have been involved. And then these are collaborators and of course our funders. So thank you very much. And I hope we can have some time to take some questions. I think you can write questions in the chat and I'll do my best to answer them.
Thanks a lot. Thanks a lot. It was really impressive from my point of view. I mean, impressive results that you have obtained with this research, and I think your talk was very inspiring. I hope so. I mean, I think you are offering a different way of thinking to roboticists, and I think this is very important, especially for the young roboticist. So I'll wait for some questions. I don't see them appearing yet, but of course I have several coming to my mind. While we wait for questions, I would like you to a bit, I mean, it's more common than a question, but to what extent embodiment is important? Of course, it is important, and we start from that. And you touched this a bit, but my question is that to what extent is not only the neural circuitry, which is also, of course, arranged physically, I mean, anatomically, in a way to make let's say computation easier, but how about the physical body? For example, the arrangement of receptors. So the eye of the ants are that kind of arrangement because I think that makes the interpretation of images easier.
Yes, yeah,
right.
No, I think that's right. I mean, as I kind of stressed, I think it's important, what kind of light it receives. The fact that it receives UV turns out to be quite important. The fact that it is an omnidirectional view seems to be important. And recently we've also been looking at this, how they detect the polarization pattern, which people have kind of, tend to assume that what you're trying to do is find what's the dominant direction of polarization in the sky, and you can kind of use that as your compass. But we've actually been considering, if you look at the actual layout of the polarized light receptors on the surface of the top surface of the eye, it looks like they're actually arranged very nicely to kind of match what the real sky pattern of polarization is going to be. And the big advantage of this is because if you imagine, if you just get the polarization directly, a single direction of polarization, it's ambiguous. It's 180-degree ambiguous. But if you actually use the whole pattern in the sky, then you can easily disambiguate that, except for unless the sun is exactly at the top point in the sky, which almost nowhere in the world is that the case. So it turns out that layout actually simplifies it. So that's something we're actually trying to do now, is to build a polarization sensor. So people have built polarization sensors for robots before, but always with the idea of just getting one dominant direction. And we're trying to build one that actually tries to extract this sky pattern and therefore gives us directly the information we need for the compass.
So the resolution of receptors is somehow balanced. It's a trade-off between efficiency and how can I say amount of information gained. I mean, if you could replace ant eyes with high resolution eyes, probably it wouldn't work, because it has to be balanced somehow with the I mean, computing capabilities, and that's very interesting. What I think is very interesting of this kind of studies is that nature can actually provide robotics with very, let's say, efficient way of processing large amount of data. I mean, the sensory, basically the sensory data coming into an animal or a robot. And I think that nature gives very elegant solutions. in the way the neural circuitry has evolved, so very efficient in computational terms. But as you just said, but what I see happening in robotics is that we don't care too much, and we just use more powerful computing machines, so more powerful computers. This is what happened in computer vision, for example, and robot vision then. So I don't know if it's a real question, but how do you see this, and why do you think that we cannot convince roboticists to use these more elegant, more efficient solutions that we see in nature?
Yes, so I mean, I think there's possibly again a trade-off that, you know, sometimes these more elegant solutions are very nice, but they're very nice for certain niche or problems. So if you want to do a certain kind of navigation, then this is really the way to do it. But maybe it won't generalize if you then, you know, the insect probably isn't any good at face recognition or whatever. Although having said that, people have shown that bees apparently could recognize faces, but we'll see. But yeah, so yeah, it depends a little bit how general purpose you're trying to be. I think that maybe pushes you more towards these solutions. And I don't suggest that, you know, insect brains will solve everything that people want to do with robotics. If you want to build a humanoid robot, then you probably don't want to try and give it an insect brain to control itself. But I think there's a huge range of applications for robots where you do want maybe, you know, more efficient processing, and you have, you know, the problem really does match the problem that the animal is doing, and that's where you can exploit it.
Okay, so I'm seeing some questions appearing. One is in the question and answer box. It's Barbara Mansolei, so thanks for the great talk. Are the tactile feedbacks important in ant navigation?
Say again?
The tactile feedbacks.
Right, yes. So for the moment, we haven't included any kind of tactile feedback. There's probably two things where it's relevant. So one is that people have shown that they do recognize the surface they run over. So I talked about this kind of, you know, Gestalt memory of what things are like. So if you change the ground surface on a familiar route, they definitely hesitate and notice that something has changed about the route. So that suggests that the tactile ground surface is actually part of their memory of the place, as well as this visual memory. A second thing is, I mean, it's maybe not strictly tactile, but it's the kind of proprioceptive information from their stepping seems to be involved in theirs. So for ants, the speed information for path integration seems to come at least partially from step counting. But they can also actually do it, they can path integrate either with step counting or with optic flow, because I don't know if you saw in my little video at the end, there was one ant carrying another ant. An ant that's being carried can also do path integration, which is quite amazing. So, yeah.
He also asks about the energy costs, if you have any estimation of the energy cost during navigation for the ant.
It must be tiny. I mean, I probably can't really put a number on it, but I mean, their brain is so tiny. The ant itself doesn't, you know, it doesn't even eat the food. It just takes the food home and comes back for more. So it must be, you know, really, but I'm sorry, I can't put a number on it, but yeah, clearly much less than any robots that we use.
The question is in the chat. So it's Paula Arena. Thanks for the impressive lecture. Can you briefly comment on the basic neural models that are embedded into your path integration model? And he's asking if there is any learning procedure and what is the complexity of the network model, and in the drone, is the model running on board the drone?
Right. So the final question, the answer is yes, it is. It's running on the drone. So here we're using the neurons. We're using spike rate. We're using rate-based neural code. So we're not using complex neurons. Each neuron is just a rate-coded neuron. The total number of neurons in that circuit is less than 100. So it's really very easy to run it on a drone or on a phone or whatever. And then, yeah, so that's the kind of neuron model that we're using. We have just recently looked at translating this into a spike model just to check that it's kind of plausible. And then there isn't any explicit learning. What there is an integration memory. So that's what you need for path integration. So at the moment, we make the assumption that, you know, there are neurons that are able to act as integrators, i.e. they get an input, but they store that input and they hold that input. And this is something that we're also looking into. So there's several candidate neurons in different systems that people have seen that have this property that you put activation into them and then they'll start firing at a particular level. They'll hold that level without any input, and then you put more input and they'll go up to another level. But to do what we need them to do, this needs to actually be quite accurate and also to have a very long time constant. And so far we haven't really found a good plausible physiological way for that to work. So that's the kind of memory in the system that we have to still understand exactly how it works. But other than that, there's no learning or adaptation in the system. It's otherwise kind of hardwired. I hope that answers the question, Paolo. Do ask some more if it doesn't. Yeah.
It's not replying, so I think the answer was satisfactory to him.
I know Paolo's done a central complex model as well, so yeah.
Yes, so okay, thanks. I'm not seeing any other. So as I was saying in the beginning, what I liked of your work is not only the inspiration that we can get from these efficient neural models, but also the way they can help understand the animal itself.
Mm-hmm.
So in your work, I see, what is the trade-off between what you know from biology and what you can add by testing the different models with a robot? So in your case, I see that you had some information on the insect brain and the different parts of the brain, and a little bit about the circuitry was known, the neural circuitry.
Yeah, so I think what's been really important for me is that I'm looking at this circuitry from the point of view of trying to solve a robotic problem, if you see what I mean. So people have known for a long time that the mushroom body does olfactory pattern learning, and there's amazing work that's coming out about the details of that circuit. But no one had people hadn't made the connection that, well, what we need to do in visual navigation is actually exactly the same computational problem. And so maybe that's where this could be happening in the brain of the insect. So it was that I had the problem in mind, if you like, so that when then I'm talking to the insect neurobiologists, I can sort of see this connection, or they can make the connection. So in the case of the central complex, I had actually presented a model, a completely hypothetical neural model that we had come up with for path integration and steering. And then Stanley Heinze, the biologist, came up to me afterwards and said, well, I think the central complex that I've been doing all these connection mapping, I think what you just showed, I think we can map those two things together. And it looks like it's the same principle. And we went through it, and it took a while, but in the end, it turned out that indeed it was exactly the same mapping. So it was, it's not that you come from if you just look at the biology that you will then suddenly realize this is what the biology is doing. You have to come at it with the robotic problem in mind, if you see what I mean, to understand, which I think is quite important. And sometimes it won't work. Sometimes there might be a robotic problem and it just doesn't map onto anything in the insect brain, and there is no solution there. But that's kind of the strategy, is to be thinking about what you can read or hear or learn about the brain from the point of view of problems in robotics, because in the end, those are the same problems, the animals have had to solve almost exactly the same problems as our robots have to solve. They have to control themselves in a complicated world with complicated sensory input, complicated motor output, and they do it much better. I mean, it's still the case that insects are much more impressive than any of our robots that were built. So.
Yeah, thanks a lot. Another point from Paula: how strong is the interplay among the MBS and the CX in the end regarding learning and memory? In the fruit fly, it is not so explicit, he says, from an anatomical point of view.
Yes, so that's an interesting question, which we're definitely looking at, is how do you put these together? And actually a few other groups have published models recently, I think just this year, of different ways in which the mushroom body stuff that I described and the central complex stuff could come together. And it turns out it could be that you don't need a lot of interconnection between them to get this to work. So we're kind of assuming the mushroom body output is this relatively simple signal about does the world look familiar or not.